Okay, thank you for all your hard work so far. Now let's dive into the cell and look at some of those intracellular structures. Remember that prefix intra means inside. So we're going to look at things inside the cell. And we are going to start with that nucleus. And the nucleus is where all the genetic information of the cell is stored. And because of that, the nucleus has the function of controlling all of the functions of the cell. It tells the cell what to do and when to do it. It's like the boss of the cell. Now, if I ask you for the function of the cell, don't tell me it's the boss. Tell me it controls all the cellular functions. Um, Inside the nucleus, we have that genetic information. Most of the time, that genetic information is free floating in that form, as DNA. This chromatin, um, that helix, double-stranded helix of DNA condenses around proteins. And at the time of cell division, it will condense and form those visible structures known as chromosomes. So sometimes that genetic information is chromosomes, but most of the time it is just chromatin, that free-floating DNA. So we're going to take a moment and look at the specific structure of the nucleus. But notice that the nucleus is surrounded by what we call the nuclear membrane. This is a phospholipid bilayer. And in the nuclear membrane, we have these openings. You see all the little like punch holes? It's called nuclear pores. And the nuclear pores are openings that allow substances inside the nucleus to exit the nucleus to get into the cytosol of the cell. Also inside the nucleus, we have this dark spot in the middle known as the nucleolus. So make sure you know how to spell nucleus and make sure you know how to spell nucleolus. The nucleus is important because it is where two different things are made. The first thing that is made in the nucleolus is RNA. So DNA stays in the nucleus all the time, but DNA is going to be used to make RNA, because RNA, as you know, is single-stranded, and RNA, because it's single-stranded, can exit through those little nuclear pores to go into the cytoplasm. The other thing that's made in the nucleus are the subunits of a ribosome. So ribosome is made of two pieces that join together. If the two pieces join together in the nucleus, they would be too big to exit through the nuclear pores. That'd be like making a fist and you can't get it out through the hole. But the individual pieces are small enough to exit through the nuclear pores. And then once they're in the cytosol, they are going to join together. So this shows the RNA exiting through the nuclear pore. And actually what is going to happen, and is that this is going to join up with the ribosome, and this is where we are going to be making proteins, as we will learn, I think it's next week. So once you're outside of the nucleus, you are in the cytoplasm. And so the cytoplasm contains lots of the solution of the cell, the cytosol. And this cytosol is almost 90% water. And dissolved in this water, we have ions such as lots of sodium, a little bit of potassium, a tiny bit of calcium, a few other things. We've got a little bit of protein, little lipids, but more of the monomers of it, for instance, glucose, fatty acids. We've got a little bit of ATP. This is where chemical reactions are going to happen in the cell. They're going to be mostly in the cytoplasm of the cell. In addition, we might have inclusions, which is where nutrients can be stored, such as chemical substance, depending on the cell type. So for instance, in your skin, If you go out in the sun, you know how your skin gets tan? Well, that's because your skin would have inclusion storing melanin. So that's an example. This is also where we are going to find all the other organelles. So we have a lot of various cytoplasmic organelles. And when you learned about them in grade school, you learned a bunch of names. So what I really want you to know is that each organelle is specialized. Each organelle has a specific structure, and therefore each one has a specific function, which means those cells whose drawings you learned, there is no cell that looks like that in your human body. Okay. The number and the variety, in other words, like I'm going to go through between a half a dozen and a dozen different organelles today. Some of your cells only have half of them. Some of your cells have 3/4 of them. Which ones a certain cell has depends on what that cell needs to do. And that depends on where that cell is in your body and what that cell is supposed to be doing. Now, a lot of these organelles are going to have a phospholipid bilayer, just like your plasma membrane, and that means they are part of what we're going to call an endomembranous system. So there's about 5 of them that are going to form this system that goes between the nucleus and the plasma membrane. So these are the ones I'm going to be talking about first. Okay, so the ribosome is not one of them, but since I talked about the ribosome, I need to finish talking about the ribosome. So here's what a ribosome looks like with its 2 subunits. And like I said before, the function of the ribosome is to make protein. So this is, oh, what type of photo is this? Go back and think about the 3 types of microscope and which type of microscope would show you this. Yeah, this would be an electron microscope picture. And so each one of these dark little balls is a ribosome. And from each ribosome, you can see a little strand of amino acids coming off. So each one of these ribosomes is actually making a protein. That's the function of a ribosome. So this actually is a transmission electron microscope because we are looking inside of our cells. Now, some ribosomes are free floating in the cytoplasm, and some ribosomes are going to be attached to another organelle known as the endoplasmic reticulum, thereby changing it to something called the rough endoplasmic reticulum. And those proteins are going to be used to make cell membranes or explode. And here's the thing about ribosomes. They don't play just one political party. They're not Republicans for their whole life or Democrats for their whole life. They go to whatever the cell needs them to be. Maybe they're going to be free floating for an hour or two. Maybe they're going to be attached for the next 3 days. They go back and forth depending what the cellular needs are. So this is that overview on one slide, what we're going to spend a whole lecture series talking about next week, I think it is. Okay, so let's talk about the endoplasmic reticulum. There's 2 types of endoplasmic reticulum. So let's see where this is. Notice right here, here's our nucleus with our nuclear envelope. And I want to draw your attention to that physically attached to the nuclear envelope. As in the nucleus is not sitting over there and sitting in the corner is your endoplasmic reticulum. They are attached to each other physically. Is your endoplasmic reticulum. And when you look at the endoplasmic reticulum, all of this is endoplasmic reticulum, but you see this part that is closer to the nucleus has these bumps on it. It looks like it has a disease. Those bumps are ribosomes attached to it. And to make it so you notice even more, they've colorized that dark red and made them very fat and flat. Whereas this part of the endoplasmic reticulum, they've made it a different color, and they've made a more round shape instead of being flat. So they realize, oh, look, that looks a little different from that. Because remember what we said? Change in structure equals what? Change in function. So let's talk first about the rough endoplasmic reticulum. It has attached ribosomes. What do ribosomes do? They make protein. So when these attached ribosomes make their protein, they dump the protein directly into the rough ER. Why? Because they're attached to the rough ER. So the rough ER basically is going to process that protein. What does that mean? It's kind of like a purse. It's holding the proteins. It doesn't do a whole hell of a lot. But what it can do then, it can pinch off a little piece with the little proteins and go, oh, goody, we're going to go for a trip. Take them like a little car. Go someplace in the cell. Take those proteins where they need to go. In contrast, the smooth endoplasmic reticulum, does it have anything to do with proteins? Absolutely not, because there's no ribosomes attached to it. So you're going, oh, goody, don't need to learn anything about. Nope, smooth endoplasmic reticulum is more complicated. It has 3 jobs. How many do you need to know? 3. You need to know 3 out of the 3. The first job, it has the function of detoxifying. So yeah, if you got a bunch of toxins, its job is to break down those toxins so you don't die. That job falls to your liver. So in your liver cells, you're going to have a truckload of smooth endoplasmic reticulum. The second job of the smooth endoplasmic reticulum is to make lipids. And this includes hormones that are lipid-based hormones. We call those your steroid hormones. You like your estrogen, you like your testosterone, you can thank your smooth ER, because without smooth endoplasmic reticulum, you would have no sex-based hormones. It's that simple. The third function is this is where the cell stores calcium. So if the cell needs calcium for something, here's where it's stored. Otherwise, the cell has to break down bone to release calcium into the bloodstream, and then the blood has to carry the calcium to the cells that need calcium. As you can see, that might be a long, drawn-out process, and maybe you don't want your bones being broken down right this second. Wouldn't you like to have a ready supply of calcium so your muscles can contract, because it's required for muscle contraction? Yeah, so your muscle cells, they need that calcium stored in the smooth endoplasmic reticulum. So 3 functions for smooth ER. Now that brings me to the Golgi apparatus. The Golgi apparatus is always described as flat membranous sacs. I like to kind of describe it as a stack of pancakes, because that's what it looks like. Just a stack of pancakes. And here's what it does. It's kind of like a UPS center in that it is going to have incoming packages that it's going to modify. So think of a bunch of smaller packages going to come in, and it's going to figure out, oh, these go together. So for instance, maybe it is going to have a piece of rough ER that came in carrying proteins and a piece of smooth ER that came in carrying lipids. And it's going to put the 2 together to make a lipoprotein. Ah, see how it did that? Put a lipid with a protein, make a lipoprotein. Or it could put glucose with a protein to make a glycoprotein. Okay, so it modifies, dehydrates packages, proteins, and lipids made in smooth and rough endoplasmic reticulum. Okay, then pieces of Golgi's can get pinched off for transport to final destination. So I've talked about pieces of endoplasmic reticulum being pinched off and pieces of Golgi being pinched off, and we don't call them pieces of pinched off endoplasmic reticulum and Golgi. We call those little things vesicles. So These little membrane enclosed sacs, whether they come from smooth endoplasmic reticulum or rough endoplasmic reticulum or Golgi apparatus, they're all called vesicles. And each vesicle only has one product type. So the lipid would come from the smooth ER, the protein would come from the rough ER. They would both go into this side of the Golgi. They would be packaged together and they would exit as a new product from the other side of the Golgi. So here they are exiting. And so what's going to happen? Well, then they can be taken to the part of the cell where that is needed. Or if it's decided like, hey, you all made a hell of a lot of this and we got more than we need, then the cell will say, okay, we're going to get rid of that. And how are we going to get rid of something big? That process of exocytosis, right? Because it's a large molecule. It's going to be exocytosis, and it's going to require energy. And so with endocytosis, as in bacteria, so what would we call endocytosis of bacteria? Eating a bacterium, what would that be called? If you don't know, go back and review, come up with that word that means to eat. When that bacteria was eaten, that would also appear in a vesicle. Okay. If it's not needed immediately, it can be stored in a vesicle. That brings us to another organelle known as a lysosome. Now, lysosomes, as opposed to vesicles, only come from Golgi, and lysosomes contain digestive enzymes. So what do digestive enzymes mean? It means it's going to catabolize other stuff. So if you take a lysosome and you put anything in with it, it's going to break it down. So for instance, if we have a vesicle, that contains food, and that vesicle merges, fuses with the lysosome, then that food particle gets broken down. So that protein would get broken down to amino acids, or that lipid would get broken down to fatty acids and glycerol. Whether a carbohydrate would get broken down into, that's right, sugar, specifically glucose molecules, so we can make some ATP. It also would happen to, like, for instance, if you have a damaged organelle, like mitochondria, it would break it down. Remember, we talked about we have a storage form of glucose, 48-hour storage form of glucose. If you're running a marathon, you want to break down that storage form of glucose, that glycogen. It's going to fuse with a lysosome to give us that glucose. If we needed to get calcium out of a bone, a lysosome is going to break it down to give us that calcium. Ladies, when you have your menstrual period every month, we're going to break down that lining known as the endometrium. So lysosomes, very important. Now, what happens if, oops, that lysosome bursts? Your cell's going to die because it's going to break down everything in that cell. And so with that, I am done talking about all the things in the cell that have this phospholipid bilayer. And so on this diagram, they all have like little leader lines to them. So go through and see if you can label them. Thank you, and I will see you again as we continue with the remaining structures inside the cell that you need to learn about. I'll see you shortly.